Greetings once again, Bio 205 students. You will be happy to know that what we are going to cover in Chapter 27 is a minuscule amount of the chapter, so this will be a very quick lecture for you. We're just going to cover a couple of topics. So, like I always say, pay attention to what we're talking about here, what's on the online quizzes that you can access via the website, and what's in the chapter outlines, because it is not going to be very much. So first topic, don't want to stop this diversity unit without reminding you that microbial diversity is enormous. They are everywhere. They do everything. There isn't a place on Earth that you can find where you won't find these little buggers making a living, because they are able to metabolize all sorts of carbon sources. They utilize energy sources that are totally different from other organisms. And not only that, they're able to move between metabolic strategies. So where members of kingdom Animalia, for the most part, are locked into a particular metabolic strategy, lots of microorganisms are capable of living for extended periods, even forever, in fermentative conditions, undergoing respiration when the conditions change, and sometimes even moving between aerobic and anaerobic respiration. In addition to that, there are organisms who are capable of growing both heterotrophically and autotrophically, so they can photosynthesize or chemosynthesize when the conditions are right for that, and if they are not, then they will move to heterotrophic growth, just like you and I, collecting organic material from the environment and consuming it. They also are members of lots of different symbiotic relationships. We're going to spend a lot of time talking about the parasitic relationships that microorganisms have with human beings later on. So here we're going to focus on the good news, the mutualistic relationships that are found between microorganisms and mammals, a particular group of mammals called ruminants, which are things like cows and other grass eaters who have this digestive organ called a rumen, and it turns out that in the rumen there are bacteria and protozoans and fungi and methanogens, and their role there is to actually take the grass, which is eaten by the cow, partially digest it into things that are digestible to the cow, and then those things move further down the cow's digestive tract. Because it turns out, just like termites can't eat wood, cows actually can't eat grass. They rely on the rumen microorganisms to break that grass down for them into manageable pieces. In addition to that relationship, we have another mutualistic relationship shown here, and that is a fungal partner that surrounds plant roots called a mycorrhizae. And mycorrhizae, as you can see from these two pictures, are really important for good growth of the plant partner. So they surround the roots and they enable them to take up more water and take up more nutrients so that the plant grows bigger and faster. What you're looking at here is two plants who are genetically identical, fed the same thing, exposed to the same amount of water, sunlight, all that good stuff, and they're genetic clones of one another, and lo and behold, one is about twice as big as the other because of that fungal partner associated with its roots. And before we leave this topic of microbial diversity, we just want to remember they live in extreme environments in addition to these more normal environments, extremes of pH, extremes of temperature, extremes of salinity, extremes of pressure. Anywhere that you can think about looking, you will find these microorganisms not just eking out a living, but doing pretty well there. All right, so that's topic number one. Topic number two is biogeochemical cycling, and we're going to look at three biogeochemical cycles. The first, which is pictured here, is the carbon cycle. Lots of stuff going on in the diagram showing the carbon cycle. What I would like you to pay attention to is the fixation of carbon and the respiration of carbon. So the fixation of carbon is just an ecological way of saying autotrophy, so the chemosynthesis or photosynthesis that takes a carbon dioxide molecule and converts it, reduces it to an organic molecule. And that here, when we consider things from an ecosystem level, is called fixation of carbon. So when we talk about the biogeochemical cycling of carbon, we're talking about the cycling of carbon between its organic form, and that's where we get the bio portion of that word, to its inorganic form, which is where we get the geo portion of that word. So fixation accomplishes the bio portion. It takes inorganic carbon and converts it to organic carbon. And we learned about two processes that do that in chapter 5, photosynthesis and chemosynthesis. In this chapter, again, because we're looking at things on a more ecosystem and global scale, we call those processes of photosynthesis and chemosynthesis, the autotrophic processes, sinking of carbon. So any process that converts CO2 into an organic carbon molecule is called a carbon sink. Likewise, the other end of this cycle, anything that takes organic carbon and breaks it into CO2 or oxidizes it, is considered a carbon source. Now, lots of things are carbon sinks, lots of things are carbon sources. We are just talking here about processes that microorganisms are involved in. And as you might have guessed, there are lots of microorganisms who are heterotrophic in nature, and so they are constantly taking organic carbon, just like you and I, oxidizing it completely to CO2 and exhaling that CO2 out into the atmosphere. And whenever we take an organic molecule and oxidize it completely to CO2 metabolically in a cell, that's a process of respiration. Remember, anything less than that is that fermentative habit where you have an organic molecule incompletely oxidized to an organic molecule, so there's no CO2 produced. So carbon sinking, taking CO2, converting it to an organic molecule. Carbon sourcing is taking an organic molecule and oxidizing it to CO2. That sinking is the one that we pay attention to here because that's our primary producers. Those are the organisms that are taking CO2, converting it to a biological form so that it's available to the biosphere, to the organisms within the environment, which we refer to as producers. And across the world, primary production accounts for more than 100 billion tons of carbon dioxide fixed into organic carbon every year. And kind of interestingly, I think, is the fact that half of that is done on land, but the other half is done by microscopic organisms on the surface of the water called phytoplankton. So plankton meaning a very small organism, not always microscopic. Some of them are visible to the naked eye, but they're very, very small, and we can certainly see them better with a microscope. And those would include the microscopic unicellular green and red and brown algae that we talked about in chapter 12. So half of all the primary productivity on the planet is done by those phytoplankton, those teeny tiny plant-like protists who are photosynthesizing on the Earth's surface in the water. And interestingly enough, if you look at this graph at the bottom, most of that aquatic primary productivity occurs in the North Atlantic Ocean, where you see all of that red. That's the biogeochemical cycling of carbon, right? So movement between fixation and respiration, which allows for carbon sinking and carbon sourcing. And that one is pretty straightforward. To be honest, this next one is a little more complicated. The nitrogen cycle, you can see from the key there, involves ammonification, fixation, nitrification, and denitrification of nitrogen. But the good news for you is the only one that I really want you to pay attention to is nitrogen fixation, which you can see there bound by the red box. And the reason why we're paying attention to that one is because the fixing of nitrogen, which refers to the conversion of inorganic nitrogen into an organic form, is something that is done only by prokaryotes. You might notice right above that red box it says leguminous plants undergo nitrogen fixation, but leguminous plants all have microorganisms living in their root nodules, and they are actually what are fixing nitrogen. So plants do not fix nitrogen. Plants rely on bacteria to fix nitrogen for them, and the product of nitrogen fixation is ammonia. So nitrogen fixation converts nitrogen gas in the atmosphere to ammonia in a process that looks a little bit like this. Up here, you have two nitrogen atoms triple bonded to one another, which means it takes an incredible amount of energy to break that bond. Notice it has to be done in a stepwise fashion where first one bond is broken and you get two NH molecules, and then another bond is broken and you get two NH2 molecules, and then finally the third bond between those two nitrogen atoms is broken and you have two individual ammonia molecules. And as I mentioned, this is incredibly expensive. Notice it takes between 16 to 24 ATP to make two molecules of ammonia, to fix one single molecule of nitrogen into its organic form. And because it's so expensive, the cell that can undergo nitrogen fixation needs a source of free energy. It either needs to photosynthesize or it needs to live in a root nodule where a plant is feeding it all the time. So you'll notice here we have three different kinds of organisms that fix nitrogen. Some are plant symbionts. They live within the plant. So this would not be like the mycorrhizae who are living around the plant roots. These microorganisms are actually inhabiting those bulb-like structures within the roots, and they live there, they're kept warm there, they get, whatever they need. They hate oxygen, so they remove all the oxygen from there, they pump sugar in, and the job of those microorganisms, those bacteria, is just to fix nitrogen. Another symbiotic relationship that can result in nitrogen fixation is a lichen, so long as the photosynthetic partner in that lichen is a bacterium and not an alga. And then the third way that microorganisms can get free energy and allow them to expend lots of energy fixing nitrogen is where we have photosynthetic bacteria who are capable of fixing nitrogen. So not as part of a lichen, but just living freely and photosynthesizing. So very expensive process, requires a lot of energy. So there has to be some kind of living arrangement where a lot of free energy is given to the cell, either because of a partner that is a plant or a fungus, or because the photosynthetic organism is just capable of producing a ton of ATP through photosynthesis. All right, the final biogeochemical cycle we're going to talk about in this chapter, even though there are two more in the textbook, is sulfur cycling, which again has sort of a complicated cycling, which includes dissimilation, assimilation, oxidation, and anaerobic respiration. But we are going to look at the relationship between sulfur cycling and primary productivity, which hopefully sounds a little bit familiar to you because we learned about this process in chapter 5 when we talked about sulfur oxidizing bacteria, those bacteria who are capable of taking a reduced sulfur compound like hydrogen sulfide, which is H2S, and oxidizing that hydrogen sulfide, taking the electrons from it, running the electrons through an electron transport chain, and producing energy, and cycling that sulfur in the process. We talked about that as a form of chemoautotrophy, sulfur oxidation, and we talked about the fact that that's common in the deep sea and also in rocks, and in either of those two places, these are actually the primary producers. You're living in an area where there is no sunlight present, so there are no phototrophs, no photoautotrophy going on. Instead, a lot of the time you have organisms taking those reduced sulfur compounds and oxidizing them in order to move electrons through an electron transport chain, translocating protons across, allowing those protons to move through an ATPase in order to make ATP that fuels the fixation of CO2 to organic carbon. And again, hopefully that sounds familiar. If not, here's a little refresher for you, right? So what do we care about with the biogeochemical cycling of sulfur? We care that we have that as a form of primary productivity in the deep sea and endolithically. So that's all for the biogeochemical cycling. We're going to switch gears now and talk about microbial decomposition, the using of microorganisms to decompose or break down organic materials. So microbial decomposition involves exploiting bacterial populations who have a normal tendency to break down organic material and have them do that in a way that is beneficial to us. So it's sort of a biotech aspect of the microbial decomposition, and it includes all sorts of really interesting things. The catch-all phrase is bioremediation, and that involves pollutant detoxification, which you can see on the upper right-hand side of your screen, where these rocks that have been covered in crude oil after the Exxon Valdez oil spill in Alaska were actually fertilized with nitrogen and phosphorus, and that caused microorganisms to come in and decompose that crude oil. And it's kind of crazy to think that microorganisms would be able to eat crude oil, but if you imagine microorganisms who are living endolithically, that's where crude oil is found, and that's their natural food source. So we've exploited that. We removed the genes from these organisms, right? Remember in chapter 9, we talked about genes of interest. We move those genes into plasmids and move plasmids into things like E. coli, and E. coli will do all sorts of cool stuff for us, like detoxify pollutants. One subset of bioremediation is something called bioaugmentation, which is particularly the bioremediation of soil or water, where you have microorganisms eating some kind of soil or water contaminant and converting it to non-toxic end products. A lot of research is being done currently to try to produce things that are more easily decomposed, and you've probably been exposed to and used some of these things without even noticing it, and they're really cool because these things that maybe look like plastic are actually compostable, and the reason they're compostable is because they're not made of plastic, they're made of plant or other materials that are easier for microorganisms to digest. So as people started studying microbial decomposition, they started asking themselves, what can we do to make it more likely that microorganisms are going to be able to decompose pollutants and other things in the environment? And what they've come up with is this idea that if you use something that's a natural product, it makes it much more likely that microorganisms are going to be able to eat that. And up at the upper right-hand side of your screen, you see gigantic polysaccharide granules inside of some bacteria. And the polysaccharide granules that are shown there are filled with something called polyhydroxybutyrate, which is a plastic-like polysaccharide that bacteria normally produce as a storage product. So if you make, say, the rings that surround your six-pack out of polyhydroxybutyrate, and then you throw those rings into your garbage, the microorganisms in your garbage will actually eat the plastic ring for you because it's not made of plastic, it's made of bacteria food. So using a natural alternative to that typically used plastic product. Another really interesting avenue of research is using herbicides that do not persist in the environment for as long because they're easier for microorganisms to decompose. You see the example here of two different herbicides with one very, very small difference between the two of them. It just happens to be a chloride or a chlorine attached to one, the red one, that is not attached to the benzene ring of the black one. And without that chlorine attached to the benzene ring, notice the relative concentration of the herbicide, even in under 2 weeks, is drastically reduced, where the red one persists at 100% concentration for an extended period of time. And the way that this can be used is to spray down and control the growth of weeds while a food crop is just getting started. So you allow the food crop to grow without being choked out by weeds, and once the food crop is better established and a little older, the weeds don't bother it anymore because it has a deeper root system and is able to outcompete the weeds. So those are some things that people have been working on and trying to figure out in order to bioremediate, right? To get rid of some of the trash that we make. The last topic that we're going to cover in this chapter is composting and bioconversion. And both of these involve the use of microorganisms to take organic material and convert it into inorganic products. So composting is a very old process. It's been used as long as people have been farming. People understood that you could put cow manure or even fish or things like that in the ground next to crops, and as those materials decompose, they would fertilize the crops. So composting is done both on a home scale and also on a municipal scale to produce fertilizers. What people recently discovered is that you can take the biomass that would normally be composted and you can, through a process called bioconversion, convert that biomass into a fuel. So bioconversion is a special kind of microbial breakdown where you're not producing compost, you're actually producing fuel in the form of methane or ethanol or a biodiesel, because you're breaking down some organic material. Maybe it's leftover vegetable oil, or it's the corn stalks from corn, and you're converting those things into a fuel that can be used to fuel combustion engines and all sorts of other really cool things, right? So biodiesel is made from the conversion of vegetable oil, which sort of enjoyed a heyday about 10 years ago, where people were going into taco shops and other places and collecting the used vegetable oil, bringing it home, running it through a filter, and then using it after just a few inexpensive modifications to the diesel motor of their cars into a fuel that would run the diesel motor. And now that people understand that that's a possibility, this is done on a much larger scale. And same thing with plant matter. They can be used to fuel ethanol fermentation that, on a small scale has been known for hundreds of years as a way of producing alcohol, but can also be used on a larger scale to produce ethanol, which, even if you don't have an ethanol-fueled car, is actually added to the gasoline that you pump wherever it is that you get your gas. And you can check that out the next time you get your gas tank filled. You'll notice that it will tell you that a certain percentage of the gas is ethanol, and that ethanol is produced by using excess plant matter and forcing the fermentation of that plant matter by microorganisms to produce alcohol. The production of methane is a natural byproduct of composting, and it's one of the reasons why compost piles don't smell very good, but people have learned recently that you can trap the methane gas from this composting operations, and you actually can use it to fuel the production of electricity. That gas will be used to boil water, and as the water turns into steam, the steam rises and it moves through turbines which spin and generate electricity. So you can see here a picture of methane gas being produced and collected from one of these giant municipal water treatment plants, and then on the lower right you can see micro turbines producing electricity from the methane that's produced from composting. So some really cool stuff going on, and again, all thanks to our friends, the microorganisms. So before we end this unit, we just want to remember they're all over the place, they're doing really cool things for us, and we have reason to be grateful for all those little guys running around the planet.